So I hope you'd agree that amino acids like this one are fascinating compounds. They're even more fascinating when you have more than one. You take two amino acids together and they can react, eliminating water by a so-called condensation reaction to give you this species here. Now, of course, as chemists, what I hope you recognize is we've got an acid species here. Usually we expect to see it more as the acid chloride, but no reason we can't have it as the acid. And an amine, in this case a primary amine, and when those react together, we've made this species, which is a secondary amide. There's the C double bond O going to the N. That nitrogen, of course, had the original C to it, plus another big R group here, we just think of as a big R group, one hydrogen, so it's a secondary amide. However, to biochemists, this is a peptide link. Okay, this amide part, C double bond O to the N, and then going off to each part or each residue, as it's called, of two amino acids is an extremely important link in the formation of proteins and many other really crucial, nice little compounds. You might recognize it too, of course, as when you studied some polymers back in GCSE. Anyway, what we've done here is we've taken two amino acids together and via a condensation reaction, losing water, we've put them together, joined by a peptide link, to make a so-called dipeptide. Okay, dipeptide is where you have two amino acid residues, and they're called residues here because, of course, they're residual from the amino acid. The acid end lost the OH, the amine end lost an H, and so now we have this dipeptide. Put three amino acids together and you get a tripeptide. And of course you can keep putting them together and keep putting them together and keep putting them together. When you've got less than 50 or so amino acid residues, you call it an oligopeptide. Typically more than 50 amino acid residues and you're heading more towards the protein. And of course both oligopeptides and proteins are examples of polypeptides, where you've got lots of peptide linkages, each between two amino acid residues. Now you could, if you really wanted to push it, of course, say that a protein, therefore, is an example of a condensation polymer. We've taken the monomeric units, which are these amino acids, and of course there's all different 20-odd different types of amino acids we can put together in these condensation polymers to make proteins, but that's really all they are, a monomer, another monomer, joined together by virtue of eliminating a small molecule, in this case water. Now it doesn't matter whether you got a dipeptide, a tripeptide, or a huge big protein. You have these long chains of peptides that we will always write so that the end nitrogen, the N-terminal end, is on the left, and the C-terminal end is on the right, because of course you've always got an amine group that hasn't reacted on one end of the polymer and an acid group that hasn't reacted on the other end of the polymer. Now there are, of course, tons and tons of permutations of peptides, and I use the word permutations because it's not just important which amino acid residues you have in a peptide, but which order they're in. Even if we just take two amino acids, there's two possible dipeptides, and I'll illustrate that by just bringing together glycine, gly, and alanine, ALA. So alanine is, of course, the amino acid that has a methyl group for the R position. Glycine is the amino acid that has an H for the position, otherwise known as amino ethanoic acid when you take your A levels. Now I could put these together so that I either have the glycine on the C end and the alanine on the N end, or the other way around, so it's the glycine on the N end and the alanine on the C end. You can see that these are two different dipeptides, two different molecules, okay, based on which one has got the free amine and which one has got the free acid. I should, of course, draw these always as the Zwitter ion form, and notice even when you've got a dipeptide, it's still a Zwitter ion because the free amine end can, of course, exist as its conjugate acid, and the free acid end can exist as its conjugate base. And always, again, just emphasizing N-terminal end on the left, whether or not you're drawing it as the free amine or as the conjugate acid. C-terminal end on the right, whether drawing it as the free carboxylic acid or its conjugate base version. Before we continue and talk about fun things about our peptides, it is important to remember that the peptide link is an amide. And of course, amides, as we studied back in the carboxylic acid module, can quite easily be hydrolyzed. Well, of course, I say quite easily. We, of course, have to reflux with catalysts. But let's think about taking, first of all, our gly residues. So that's the alanine is the N-terminal end and the glycine is the C-terminal end. And think about how we can hydrolyze this when, of course, we do it under reflux, either with an acid catalyst or a base catalyst. Well, if we reflux it, regardless of whether we have acid or base in there, we're going to break that amide linkage, that peptide linkage. Okay. Now, if we do it in an acidic conditions, of course, then everything we have left afterwards is the acidic form. So we take the alanine, for which the N was already in the free conjugate acid term, and then we break it here. So we got the C double bond O as part of the alanine. Well, then we put just the OH back on there, and we've got our free acid there. And then, of course, the glycine end, well, that was originally in its conjugate base term. Well, of course, when we put it in acid, it goes back to the acid form. The nitrogen's going to get cleaved, is going to be hydrolyzed, another couple of hydrogens put on there, and as we're in acid, it will turn from the amine form into the acid form itself. So reflux this with acid, and we get both alanine and glycine in their acidic forms, and of course refluxing it with base will end up giving us everything in the base form. So therefore the alanine end, this acid, conjugate acid end, will return to the base end. Cleaving it here would give us the carboxylic acid, except we're in base, so we're going to get the conjugate base version. And then, of course, as far as the glycine end is concerned, we've already got the conjugate base of the acid. When we cleave here, we'll make the amine, which of course stays in the amine base form. So we can cleave our peptides quite happily, refluxing with acid or with base, and this is an important analytical technique, if you like, something that's very important to be able to do when you want to study exactly what type of amino acids you have in a longer peptide. Now I should note that I reflux there the gly version of our dipeptide. How about if I was to hydrolyze the gly-ala isomer? Okay, so here the glycine is the one on the N-terminus and the alanine is the one on the C-terminus. Well, I hope you appreciate that I'm going to get exactly the same products. I'm going to hydrolyze again, cleave at this amide bond, and I'm going to make glycine and alanine. Again, exactly the same products, whether I do it in reflux in acid or reflux in base, exactly the same products as when I hydrolyze the gly. So different peptides have different orders of the amino acids in there. Once you start hydrolyzing those peptides, you can identify which amino acids are there, but you don't know the order they were in the original peptide. And although we're not going to dwell very much on peptide and protein analysis, in fact, we're not going to dwell at all on peptide and protein analysis. That is something important to know generally, that hydrolysis as a form of analysis is a destructive type of analysis. You might get some information out, but you lose information in that destructive part of it. Before we do leave our peptide consideration and move into proteins, I do want to emphasize something about this peptide link that is going to be very, very important as we talk about the structures of polypeptides, otherwise known as proteins. So let's just pull out that peptide link there. Okay, so the C double bond O, there's the nitrogen with its lone pair. Here's one amino acid residue going off here. Here's the other amino acid residue going off there. And of course, what I hope you recognize in this amide linkage, and of course it's something we talked about quite extensively back in the carboxylic acid module, is that you've got pi electrons here in this carbon-oxygen double bond. You have a lone pair of electrons in this nitrogen in a p orbital, and p orbitals can be overlapping quite happily in this nitrogen carbon oxygen threesome of atoms. And of course the orbitals are not just overlapping nicely, but they're all a comparable size. So what that means is that we can spread out electron density quite a bit outside of this one resonance structure. So we could move electrons from the pi bond onto the oxygen, giving us this polar species. Now we've got a carbocation there because it lost some of those pi electrons. The oxygen's got that excess of electrons. Well, of course, the nitrogen here has the lone pair of electrons in the p orbital that it can quite easily overlap with the carbon p orbital to give us a pi bond there. Okay, and of course this is that carbon nitrogen bond of the peptide link. In other words, this peptide link has got quite a bit of double bond character. And what that means is that while you think about it like this, that's a single bond, lots of rotation available. You'd think of one peptide residue whizzing around this bond, another peptide residue whizzing around that bond. Because it's got a lot of double bond character, that rotation is significantly hindered. In fact, it's not a bad idea to think of this peptide linkage almost as being a double bond in that regard. Okay, and so what that means is that the amino acid residues are kind of frozen in space around that particular bond. Now, usually the arrangement almost all the time, just simply to keep things out the way and from clashing into each other, the arrangement is a so-called trans as far as the carbons are concerned. Here's the carbon of one amino acid residue, comes into the peptide link, there's the carbon of the second amino acid residue, and it's on opposite sides of that pseudo double bond. Okay, so that's the trans alignment there of carbon from one residue through the carbon nitrogen pseudo double bond into the carbon of the second residue. Somewhat confusingly, because of course back when we did alkenes and even the cycloalkanes, I introduced cis and trans for the cycloalkanes. I pointed out it has the same meaning in alkenes, but it was easier to start using E and Z for alkenes. Well, have a look at this arrangement here, which is trans for the carbons. It's actually rather confusingly Z around the double bond, because on this carbon the oxygen has the priority, on the nitrogen the carbon has the priority, but it's the trans arrangement of the amino acid residues that you need to remember. Now, I said earlier when we were going through the types of amino acids that there are a couple of special ones, glycine and proline. Okay, well, glycine and proline are rather special in terms of this too, because glycine only has a hydrogen there. It actually means that even though the carbon-nitrogen bond has quite a bit of double bond character, it's relatively easy for glycines to rotate around because there's hardly any steric bulk with glycine. And then proline, of course, I said was special because it was in that funky little ring thing where the carbon, the so-called alpha carbon, the central carbon of the amino acid, is part of the same ring as the nitrogen. Well, when you have proline involved as part of a peptide link, 90% of the time proline is in the trans arrangement as far as the carbons are concerned. Okay, there's the peptide link, the pseudo double bond there, and you see the carbons are on opposite sides. However, about 10% of time proline is in a cis arrangement. Again, look at it here. There's the carbon-nitrogen pseudo double bond. Here's the carbon of the proline. Here's the carbon of the other residue. And you can see they're on the same side of that pseudo double bond in a cis arrangement. And this enables proline, proteins, to utilize proline to kind of change directions, going from a trans to a cis type of arrangement. But more of that in the next movie discussing protein structures.